So I'd like to talk briefly about what I call a constellation of miracles, the miniature city that we call the cell. Now, Michael Denton summarized this very well in his work back in 1986. He says, although the tiniest bacterial cells, although they're incredibly small, each is in effect a veritable micro-miniaturized factory containing thousands of exquisitely designed pieces of intricate molecular machinery made up of 100 billion atoms, far more complicated than any machine built by man and absolutely without parallel in the non-living world. The simple cell. All of us in school probably saw a textbook that had a little diagram of what they call the simple cell. It turns out there isn't such an animal. The simplest cell is more complicated. It's beyond our imagining. It has a central memory bank, it has assembly plants and processing units, it has repackaging and shipping centers, robot machines in the form of protein molecules that consist typically of 3,000 atoms in three-dimensional configurations, and there's hundreds of thousands of different kinds of these machines that make up the composite. And they communicate with elaborate communication systems that we have yet to understand. And they also have quality control and error-correcting mechanisms. Now, if we were going to make a model of a simple cell, let's assume we decided to embark on a project to build a model of a cell 1,000 million times larger than life. Well, that would make each atom about the size of a tennis ball. We would need 10 million atoms, about 10 to the 13th. That's quite a number. If you had to count them at one per minute, it would take you 19 million years to count them. That's a lot. And this model that we would make would be over 10 miles in diameter. You get a feeling that this thing is big and complicated? Absolutely. Now, we speak of the cell, we glibly make little diagrams saying that, well, it's surrounded by a plasma membrane. What we fail to realize is that in that membrane are gateways with special signal receptors and security guards to monitor what goes in and goes out. The center of the cytoplasm, of course, has as the core, it's its nucleus, which is its information center, which includes a master library with which everything is coordinated. Inside that, we have automated factories and product manufacturing facilities. All this is powered by power stations. The mitochondrions are power plants and the energy sources for this miniature city. And they have a bunch of things they call the Golgi apparatus that process, package, ship and prepare for export the various products. And they also have vesicules which transport and take care of trash disposal and all that sort of thing. Now, we have this thing is populated with robot machines in hundreds of thousands of different types, as I said, and they communicate by means of digital languages and decoding systems, memory banks for information storage and control systems for regulating all this, and indulges in prefabrication and modular construction. It has proofreading and error correction devices for quality control. Now, as some of you know, I spent 6 years of my executive career at the Ford Motor Company. At Ford, we were very proud of our very unique facility in Dearborn called the Ford River Rouge plant. Now, this is a plant that's the largest integrated manufacturing facility in the world. It has about 100 miles of railroad within the plant. Under one roof, they receive raw limestone, raw iron ore, and coal go in one end. They manufacture under that roof their own steel, their own glass, their own paint. They have an automated plant that builds the engines. They have a manufacturing line for all the rest. They assemble the mixed models, different cars of different kinds, of different colors, as you all know. And anyway, the point, the raw materials go in one end, new cars come out the other. It's considered the largest integrated manufacturing facility in the world. The reason I bring it up is the cell, your simplest cell, is more complicated than the River Rouge plant. And your cell can do something the River Rouge plant can't do. It is capable of replicating its entire self within a few hours. And by the way, all cells derive from previous cells. We know of no case where a cell comes from a startup. All cells derive from a prior cell. Think about that. Now, there's a choreography that I can't resist including in this briefing to give you a flavor of what goes on every hour or two in every cell of your body. And that's a choreography that's absolutely breathtaking, the choreography of the chromosomes. Mitosis is the splitting of the nucleus, cytokinesis is the splitting of the cell itself. And what you have in the cell is all the chromosomes condense and the nucleus will dissolve. Then a strange mitotic spindle starts to form. Now you ask yourself, who's orchestrating this? Nobody knows. How do they all know that they stop what they're doing and prepare for this dance that's going to go on? The mitotic spindle starts to form. All the chromosomes have duplicated themselves into pairs and await the rest of the dance. And we'll go into the next phase, the metaphase. As this mitotic spindle, notice they don't connect. Each mitotic spindle knows exactly where it is supposed to connect on one of the kinetochores of the chromosomes. The question is, gee, how do they know where to go? How can they see where they're supposed to go? Who's coordinating all this? Nobody knows. This whole dance, which is essential to life, of course, got all designed by chance. This is all the result of a random accident. But I want you to notice that this mitotic spindle finds its location when it finally gets connected to all of these various chromosome pairs. We're ready for the next phase. And I'll take the next two phases together to save time. What happens now, that you notice the nuclei have dissolved. How does it know that these miniature cities have decided to dissolve? The mitotic spindle pulls apart these pairs, so now there's a total duplication of the chromosomes available. They migrate to the opposite ends of this cell, and somehow this is all being coordinated, coached, whatever, quarterbacked, if you will. And pretty soon what happens is two nuclei start to form. I don't know how they're supposed to know when to do this, but it's all tightly coordinated. And you'll see the two nuclei form. And then a group of microfilaments start to pinch it off in the middle. And obviously you're about to see two cells. And so cytokinesis has been completed. One cell, you now have two exact duplicates. Now what's strange is that this exact duplication continues for a while, but then later something even stranger occurs. But before we get into that, I'd like to talk a little bit, we speak glibly about these chromosomes. I want you to imagine this assignment. Imagine that you had two strands of monofilament fishing line 125 miles long. Can you visualize that? A pair of, okay. What I want you to do is make an arrangement so that you can pack that 125-mile pair into a basketball. And how would you do that so that you could untangle it, read it, and put it back without tangling? I want you to be able to unzip it, copy it, restore it on spools at 3 times the speed of an airplane propeller without tangling. This all goes on continually in your body every time that somebody on the factory floor needs a copy of the master record to tell it what to do. The DNA is unzipped, a copy made, a working copy for the factory floor. This occurs all the time, continually. And it all happens. Now, if we take the chromatin and unravel it, you then get what they call the condensed chromatin, and you unravel what they call the loop domain. As you unravel that, you discover it's made up of a whole string of spools, little tiny nucleosomes, as they call them. And around each one, of course, is arranged what you and I have come to recognize as the famous double helix, or the DNA itself. Your DNA is about 6 feet long, and it's all wrapped up in a microscopic location. Now, most of you have seen computer models or representations. I want you to notice this double helix of the DNA consists of like a ladder of code pairs. There's an alphabet of 4 letters, always used in pairs. 3 out of 4 of these code pairs will define any of the 20 amino acids that make life possible. This is an error-correcting digital code. If you know anything about computer design, you know that couldn't happen by accident. It had to be very skillfully designed. See, life is digitally defined, and that's what Genesis says in Genesis 1:25. It says, God made the beasts of the earth after his kind, the cattle after their kind, and everything that creepeth after his kind. They are digitally defined. They're not analog, they're digital. What do I mean by digital? It's like a watch. If you have a watch with hands, that's analog. If you have a watch that says, you know, you have 39 seconds left in your talk, it's a 3 and a 9, you know. Those are digits. They take their meaning by arbitrary definitions. They're designed. They don't happen randomly. Now, see, the ultimate technology are thoughts expressed in language. You need to discern the difference between the technology of content and the technology of conveyance. There's a difference between the technology to get a radio signal to your home and a television set and the technology that may be involved in the play or the software that you're watching, if you will. The manuscript versus the media, if you will. Did the ink write the book? You can talk about binding paper and ink. It's got nothing to do with the technology of the manuscript itself. Is it in German? Is it in English? Is it whatever? Is it music? Is it text? You follow me? Totally different technologies. Now, the code of life, of course, is the DNA, and I'll just zip through this. We've just recently mapped the 3.1 billion letters in the human genome, thanks to the competition between the private sector and the government consortium. And they're 5 years ahead of schedule. They're expecting a gold mine of medical breakthroughs through all of this. But the frontier science really isn't microbiology, it's information science. They need to study coding systems that are more sophisticated than most of the cryptography that occupies our National Security Agency. And the element of language, of course, includes semantics. What do the symbols mean? Remember Paul Revere's ride? The simplest example of a signal, of course, is Paul Revere. Remember when you watched the Old North Church? One if by watching the lanterns, one if by land, two if by sea. The first bit of information carried no information. It was just proof that the guy didn't fall off the ladder. He got up there. Right? It's the second bit that told him that that's where all the information was. And it also took syntax. Those lanterns couldn't be anywhere. They had to be in the Old North Church. The position is important. Now, the most advanced computers, if the British had great computers, they could not have broken that code because it's got nothing to do, it's all took its significance by an arbitrary predefinition. If I wrote on the blackboard, M-A-N, what does that spell? Man. Why? Because we've agreed on that beforehand. It didn't happen randomly, it happened by very skillful design from the beginning. So anyway, let's move on. We have simple alphabets. You can make an alphabet that's error correcting. We do that in computer. We call it parity checks. You can make tests on your code to see if there's an error. Some of those tests can be such that not only tell you if there's an error, they can tell you where the error is. They can be error correcting. If you've ever had an opportunity to see the Super Sage machines, the solid state air defense systems, they're built on an 11-bit byte in which you can pull the cards out of the computer while it's running and it keeps going because it error corrects. And it's possible to do that. And you, of course, also have adaptive coding where codes change depending on the needs. Those are very sophisticated, but we find them in the human cell. Now, the building blocks of life, of course, are amino acids. There are hundreds of amino acids, but there's only 20 that are responsible for all of life in the universe. And by the way, they're all left-handed. Mathematically, they're a molecule that's left-handed. If they occurred randomly, you'd have half left, half right. But the right-handed ones are lethal to the left-handed ones. They always joke about, talk about the monkeys in the typewriter. If you had enough monkeys typing for 1,000,000 years randomly, you'd eventually get Shakespeare's play or something. That, of course, is nonsense for lots of reasons, but how far would the monkeys get if every other key in the typewriter was lethal to the monkey? You see? And most proteins are a linear sequence of somewhere between 100 to 500 of these amino acids, and they're of different kinds. The actual molecular diagrams are complicated, but there are certain patterns that allow you to really follow what's going on. There are 20 amino acids that make up life. Half of them are non-soluble in water. Half of them are soluble in water. The non-polar ones, the non-soluble ones, form the core of it. The other ones give it its external characteristics. And some of those are positively charged, which makes it basic, and a couple of them are negatively charged, which makes it acidic. And by these properties and by the sequence they're assembled, it causes that chain to fold up to become a three-dimensional machine to do whatever it is supposed to do. Now this alphabet is a 3 out of 4 digital code, and I won't get into all the details except to just visualize it as it usually goes by 4 letters, A, T, G, or C. That's the alphabet they deal with. And it's interesting that they always come in pairs. Whenever you have an A T will naturally bond with it. Whenever you have a G, a C will naturally bond with it, and vice versa. So if you have a string of these codes, their complement will intrinsically join it, and that gives it a self-replicating capability. Because if you tear this apart, its complement will join it, and you end up with a code that intrinsically can copy itself without error. That's the way the thing can split without errors. Now, the DNA that you have is your master file. That's your master record in the central library. You make a copy of that when you need to do something called the RNA. Visualize that like a photocopy. And that gives you, that you send that to the factory floor to manufacture these robots, these proteins that make up the city. Now, the Crick dogma always assumed, for lots of reasons, that you go from the master record to a copy, and the copy goes to the floor, and if you have corruption in the copy, fine, that can be repaired. One of the disturbing discoveries in recent years is that you have it able to go the other way. What happens if you took your master record, went to a Xerox machine, only to discover that occasionally your Xerox machine would change your master record? Would that make you uncomfortable? You bet. And that's what happens when they discover what they call a retrovirus, which has the capability to go uphill, to go back and reach in and change the master record. That's what's behind the problem with HIV and all of that. Now, when you take a DNA and copy, here's something else interesting. When you take DNA and you make an exact copy, it turns out you don't really want the exact copy, you want an excerpt from it. And so what the robots do, they extract the parts you don't want, which are called introns, and they reassemble what's left, called exons. This is exactly the kind of coding that we discover in the biblical text. There it's called the equidistant letter sequences, where every seventh letter spells out something that's relevant to what's going on, and so forth. If you've heard about the Bible codes, it's the exact thing that's going on in your DNA as we go to get the messenger RNA.